Hello, everyone. Welcome to Foresight Space Group. Today, we're really lucky to have Erika DeBenedictis, and we're joined also by Creon, who will be interviewing her. She is the CEO at Pioneer Labs. It's a nonprofit that engineers microbes for Mars. And Erika, we saw you a couple months ago where you joined our AI for Bio workshop, where you presented on AI automation for accelerating scientific discovery. But today, we're going to be talking about green Mars, humanity's climate positive future in space. So without further ado, we'll open up the interview floor with Creon and Erika. Thank you guys so
Yeah.
much.
Okay. Thanks, Lydia. And by the way, if people have a burning question or they don't understand something that is said, I think you should interrupt. I would wait until a little farther downstream to interrupt with like major questions or new topics, but there will be time for about halfway through. So thanks for coming, Erika. Before we get into the exact title topics of your talk about which Lydia just mentioned, perhaps you might tell us just a bit about your background and the various things you've done that have landed you in this place, and maybe a little about any other things that you're currently involved in that might be overlapping.
Yeah, sure. So I have an interesting sort of multidisciplinary background. So I actually started off in science in computational physics. So I did sort of like computational physics, astronomy stuff. In undergrad, I was at Caltech and I got to work at JPL doing some like mission design stuff. And so I was introduced to the whole world of science in this field where math works very well. So you can like plan orbits to Mars that are like pretty accurate with just Newtonian physics, right? The solar system's like pretty easy to model because it's mostly vacuum, right? At least at that scale. And that was really attractive to me as like a young scientist because I wanted to do cool projects and I didn't know that much math, right? I had a friend in undergrad who at one point suggested to me that maybe instead of simulating the solar system, I should try doing computational physics as applied to proteins. And so I went and did this internship at D.E. Shaw Research, which is a private research company that was doing molecular dynamics. And I was shocked by how hard it was and how bad, at least at the time, we were at predicting how biological systems would behave. And I honestly became deeply paranoid that everything I had ever simulated was crap. And as a result, I became an experimentalist and I wanted to know where real data comes from. And I wanted to know, if you can compute something or simulate it or try to predict how it will behave, how do you know when you've succeeded? Like, how do you actually check? And so during my PhD, I switched all the way from computational physics to experimental biology, a PhD at MIT that was focused on protein engineering with robotics. So I was sort of doing I had seen this protein engineering task at D.E. Shaw Research. We were doing molecular dynamics, trying to simulate proteins. And I wanted to do the experimental version of that. I didn't believe the computer anymore. So I wanted to actually test out proteins in the lab and see how they work. And so I did a PhD that was, because of my background, I had a bunch of experience with sort of traditional engineering and robotics and just coding and stuff like that. And so I walked into the wet lab at MIT and I saw that they had these very expensive, very sophisticated liquid handling robots that people were programming with like drag and drop GUIs. And I was like, what are you doing? Like, have you not heard of scripting? Like there are tools that are here to help you, right? Like we could accelerate experimental biology enormously by like just taking advantage of all the tools we already have in computer science. And so a big part of my PhD ended up being creating this system called PRANCE and robotics assisted near continuous evolution that basically a demonstration that if you use to both do directed evolution experiments in high throughput and also babysit them. So take constant measurement and make adjustments to the environment in real time, like make a simple feedback controller. Those two things together make directed evolution work better and faster, which is great. That's a very powerful engineering tool, but a downside of evolution is it can take millions of years. Yeah.
So I can see what's happening here. So you did some engineering in aerospace and astronomy, then you pivoted into biology and started doing directed evolution. And now, right, you've merged them.
Yeah,
Okay. Yeah.
that's exactly right. So When COVID hit, I had plenty of time to sit and think deeply about the world. And I sort of tuned back into what was happening in the whole field of space science. And I was struck by sort of two things. Number one, when I was at JPL, it's like SpaceX was still a joke. Like, I think in that era, it sort of felt like NASA felt very self-confident and didn't necessarily see the value of some of these new private space players. And by the time COVID happened, that was not true anymore at all. Like, the whole field of space science, I think, has become really interested to hear what these new organizations have to contribute. And they're contributing a lot, which is super exciting. So I think the field is open to new ideas. And then number two, the other thing that struck me was, now with a biological engineering PhD, and I ran an academic lab on a similar topic, I know all this stuff about what biology can do for us and what especially biomanufacturing is capable of. And I was just like shocked to see how little that is used in space science. It's a fantastic in situ resource utilization technology, but I think for a lot of reasons, the whole space science community hasn't picked it up. It's a very different type of engineering than sort of mechanical engineering, which is like sort of the bread and butter historically of like the discipline. And people don't necessarily don't think of biology as like a mission critical technology that we absolutely need, when we go to Mars, which to me, it seems obvious that that's necessary. You can't get the math to work just arithmetic wise about like how much stuff you need to take to Mars to get a human mission to work unless you're really meaningfully producing things on site. And biology is the sort of obvious way to do that. I wanted to see what I do, and that's how I got here. Yeah.
So although the aerospace and space science communities may be a little bit slow to appreciate what you appreciate, the astrobiology community certainly appreciates it and overlaps with your interest in some way, correct?
I found this really interesting, actually. So when I was in grad school, I did a bunch of work on genetic code expansion, which is adjacent to sort of origin of life synthetic biology, right? So it's the study of, why are there 64 codons? Why are codons 3 bases? Could codons be a different number of bases? That was what my thesis was about. What are the limits? Can we adjust how life works? And by doing that engineering, can we understand what is essential about life, right? This is like an origin of life. How crazy could it be? And what I found interesting actually was that the astrobiology community, I went to UpcyCon, which is like their main conference shortly after I sort of got back into space science. And I was really interested to see that actually that at least academic community doesn't talk that much actually to the traditional synbio community. So they go to different conferences, they like kind of use different vocabulary in some places, despite the fact that, yeah, there's in principle a lot of technical overlap, right? And I think a big perspective difference is that astrobiology, it's like, early on, we wrote this review paper about extremophiles. And one of the points we made is that I think the astrobiology community, they often talk about organisms surviving in a extreme environment, kind of with the same language that they talk about organisms like thriving in that environment. There's not a huge distinction there. But to me as an engineer, that is hugely different, right? There's a huge difference between an organism that can reproduce quickly, be metabolically active, like actually do chemistry in an environment, and an organism that has like sporulated and is like not doing anything, but is like technically alive. And technically, if you took it back into more permissive conditions, it could grow. And so I think the astrobiology community, yes, they're like the closest equivalent. But what I'm doing is like applied astrobiology.
The way this reminds me of something.
Yeah.
I want to take two brief tangents. The first is related to what you just said, and the last one is just a brief rewind. Are you familiar with the Surveyor 2 story?
No, tell me.
So when Apollo 12, the second crewed mission to landed on the moon, they deliberately landed kind of as close as possible to Surveyor, I think it was Surveyor 2, which was a unmanned probe that had landed on the moon a year or two earlier. And it had, various it wasn't a rover, it was just sitting there, and it had various instruments, including a camera and solar panels and all these things. And so they walked over to it, literally, and they unbolted a bunch of pieces of this thing, the camera, a solar panel, maybe some other things, and they took these pieces back to Earth with them when they returned to the Earth, so that the engineers could look at stuff that had long duration exposure on the moon for like a year or two. And one of the interesting things they found was that in the camera, there were these rubber or some kind of gaskets, like non-metal gaskets for some purposes. And in those gaskets, there were bacteria that had they hadn't so carefully sterilized this thing when they went to the moon. And so these bacteria had been sitting on the moon for like a year or two, and they brought them back to Earth. And of course, they just woke right up. So that was kind of an early version of that sort of survival mode thing, because you can be sure they weren't multiplying, right?
Yeah, and I think this is a really interesting point. And this gets into a ton of like the ethics of planetary protection on Mars and so much stuff. But, you know, even on Earth, we have environments that are so extreme that stuff just like doesn't grow. Like we have water-based life. And if the water activity of the area you're in is just below what's possible, like stuff can't grow. So like,
yeah, but some of it can survive.
But some of it can survive, right? And this is, I think back when we did the moon landings, that was when planetary protection was first written. Like at the time, it was possible that we would get to the moon and it would be covered with plants. That was like a, you know, maybe a remote, but that was possible. And people were worried about invasive species.
Well, and also around that same time or shortly thereafter, we sent the Viking landers to Mars specifically to check for life, and they had to be seriously sterilized for not only planetary protection, but not to mess up their own instrument readings.
Totally.
Okay. Let me rewind to one other thing. I hope I don't lose people here, because this is a thing I need to ask you, since you worked on classical protein folding using molecular dynamics, and then we'll get back to Mars and space and extremophiles and all that stuff. I just have a question about protein folding prediction. And I'm not talking about like AlphaFold and these kind of just pattern matcher systems, but like protein folding based on molecular dynamics or molecular mechanics, or even more advanced like ab initio or reactive models. Okay. I always thought that a great deal of protein folding in the human organism, I mean, it's not only driven by water and all the stuff that people would rather ignore, but there's all kinds of post-translational activity that happens to proteins. They get processed by various organelles and enzymes that presumably fold them in various ways. How can that possibly ever be accomplished using a molecular dynamics approach?
I mean, it can. I mean, I think people have like kind of largely given up on molecular dynamics as like the right avenue to study protein folding, simply because the time scale on which proteins fold so vastly exceeds the timescales on which we can accurately simulate proteins that it's just very tough to do it that way. And yes, AlphaFold is pattern matching, and maybe slight backstory. So after my PhD, I actually did a postdoc in David Baker's lab right when AlphaFold 2 was coming out. So that was like the moment to be there. And I was there. It was really cool. And it is worth, like, yes, it's just a pattern matcher. And AlphaFold 2 is the first computational model we have in biology, period, that can predict the result of an experiment, like, as well as doing the experiment. So like the error bars on the positions, when you do a physical crystallography experiment, you still get error bars, right? And AlphaFold gets within those error bars.
That's fantastic. So thanks for that tangent. We could talk about this, you and I, at some other point, because this is not
about... maybe one more sentence on that. It's like, you know, the fact that, recall, like I got into biology because I was worried that like computers are not going to help me in this discipline, right? And so the fact that AlphaFold works a little bit restores my faith in computation. And so it's, you know, I think machine learning in general has like become this computational paradigm that seems to play nice with like the underlying structure of problems in life science and the types of data that we have. And so using machine learning is something that, you know, may assist us in making microbes for Mars, which we could talk about later. But it's back on the table.
Let's get back on the main track here. Microbes for Mars and green Mars and all this kind of stuff. So why don't you just brief us on that thread, which I presume is the current one that you're focused on, or one of them, and not just, we kind of know how you probably got there, but what have you been doing? What have others been doing?
Yeah, so I started a focused research organization style nonprofit called Pioneer Labs, which is aiming to make pioneer species for Mars. So in ecology, a pioneer species is like the first to repopulate an area that has been made sterile from a forest fire or a volcano or something like that. And often these are organisms, like the very first ones are the ones that convert rock into dirt. Right? Take raw materials and turn it into something that higher order plants can live in. And so that's similar to the approach we're taking. So step one, if we want to have biotech be useful on Mars, or long term, if we want to farm or garden or terraform, stuff needs to grow in the dirt. So what we're doing at the moment is we are trying to create sort of like a first meaningfully useful proof of concept bioprocess. So we want to find a microbe that can grow on Mars water plus dirt, and that's it, and Mars atmosphere. And we want that organism to be part of a bioprocess that produces something useful. Likely it's going to be bioplastic that you could use to make greenhouses. And together, the idea is that we could make basically like a refrigerator-sized lander unit that you put Mars water, Mars dirt, and Mars air into, and out comes bioplastic. And that's something that we could, in principle, flight test like in the 2031 launch window. So like actually have there be a first meaningfully useful use of biotech on Mars.
It's very funny that you'd be making greenhouse plastic because, as you probably know, Elon Musk's initial goal, way when he was much younger and before SpaceX, was to buy a rocket from someone else and fly a greenhouse to Mars and grow something in it. Now, this would be something in an Earth terrarium kind of thing, but it's funny coming full circle. Let me ask about the perchlorate problem. Is the idea that this reactor would just wash them away or something like that before it made dirt?
A two-part answer. Number one, we've created organisms that can tolerate the perchlorate. So maybe you don't have to get rid of it. So Mars dirt is really interesting. By Earth standards, it's like actually has a lot of nutrients in it. It's like pre-fertilized. So it actually has a lot of fixed nitrogen in it, which is normally what we add to dirt as fertilizer on Earth. The trouble is that it also has a bunch of toxins. Perchlorate is one. It also has really high salt content, which is not great in general. So it creates sort of like osmotic stress. And it also has a lot of like heavy metal oxides. So you get heavy metal ions like into water when you add water, and that's also not great for most microbes. So Mars dirt actually has all of the nutrients that many microbes need to grow. It's just they have to overcome the toxins that like come with it. And so this is where we're sort of getting in there, doing some combination of like choosing the right organism to begin with, and then engineering it to have better tolerance to the toxins. So that's one answer. You could just choose a microbe that doesn't care about the perchlorate or consumes it. So that's the second option. You can use a microbial process to remediate the perchlorate and have it go from a pretty substantial amount to sort of below detection limits, totally safe for human and all of that.
Okay. Oh, Luke had a comment about perchlorate. What does it say? You want to just say it, Luke?
Oh, sure. So, I mean, like, I'm absolutely sure that Erika already knows this, but there's two microbes that we know about at least that can metabolize perchlorates. They're unengineered; they're just natural. One's a natural extremophile, lives in hot vents, I think. But the other one's Firmicutes, which lives in your guts. So, like, if we ever do go to Mars, we could literally just accidentally bring the solution to the perchlorate salt like with us and just have it as a bio waste product.
You just bury your poop, and there it all is. Yeah, right. Okay.
But also, like in the metabolism, it releases O2, which is like one of the only processes that releases O2. So that's nice.
Yeah, there's a whole bunch of perchlorate reducing microbes. There are consortia that do this. So perchlorate is like a rare contaminant on Earth. So there are natural organisms that do interact with it. It's a ubiquitous contaminant on Mars. It's a ubiquitous toxin on Mars. It's basically generated by UVC hitting sand. So on Earth, we get a little bit of it in like high altitude
Where
deserts.
does all the chlorine come from? It just native on Mars and other forms?
Yeah, I mean, we have it here too, right? So there's natural organisms that do it. We actually did some math trying to figure out, you know, if you wanted to make there be a thriving biosphere on Mars, the way we have on Earth, you entirely rely on energy from eating perchlorate. So some of these organisms, some can degrade it, some can actually get a little bit of energy out of it. And the answer is there's not really enough perchlorate to like have that be the only energy source. So like you have to have there also probably be photosynthesis if you want to do this. And perchlorate photosynthesis is like much better evolved than perchlorate utilization, obviously, right? Like evolution has spent a few extra billion years like optimizing it. And so eating perchlorate is not as efficient as it could be. You could maybe do some enzyme engineering, but overall, probably we'll just remediate it and then call it a day.
Okay, so Micah, do you have a quick one or do you want to wait till after?
Yes, a quick question on the same topic. You mentioned that you'd have Mars dirt, Mars air, and water. Is the water reused or is it consumed? And if it's consumed, what is the limiting factor that requires the water and we can't replace it with something else? Is it just because biology on Earth uses water everywhere and so it's simpler to use water? Or is there some other reason that makes water absolutely required?
Yeah, life uses water. So we don't know of any examples of living things that use any other solvent, right? There were some super cool experiments in the 70s when all the cool science happened where people took heavy water and they evolved some organisms in heavy water and even heavy water. So it's still water. It's just a different isotope. Even heavy water causes like fitness defects, like things will grow less quickly in it.
Mice will die. Mice will die eventually if you just give them heavy water to drink.
Totally.
Yeah.
Microbes less so. So they managed to make some algae that do pretty well in it. But
okay, so look, enough.
And the water you can reuse it. So
water is only used really for three things, as I understand it, in biology on Earth. And let's focus on plants for a moment. It is used as this universal biological solvent in all tissues. So that water has to be incorporated into the plants and stay with the plants until the plants themselves die or are harvested. Water is, of course, presumably some of the hydrogens in water are processed are transferred to other molecules in the biosystem, of course. And then the water is like the whole plant. They have this transpiration process going on, most of them. And there, the water is not used up at all. It's just flowing through the system. And
right, it's the circulatory system for a plant. It's just water. One note on this though, water. So look, I mean, it's maybe in theory you could have life that uses another solvent, but we don't have that, and that's like above my pay grade. And I think about terraforming Mars, right? Like that's real science fiction, is having other solvent.
That's funny. I like that. Okay, above your pay grade, that would be like God or something like that.
I mean, yeah. Wait, two things though. Just to have biomass is usually between like 50% and 99% by weight water. So like when I say solvent, I mean like you need a lot of water to make biomass. It's mostly water. And then thing number two, the thing you mentioned hydrogens get transferred places. Yes, that's right. So photosynthesis, the way photosynthesis works is you take a molecule of water, you split the oxygen off, you just throw the oxygen away. So this is why photosynthesis generates oxygen. And then you store those hydrogens on sugars, and that's how you store the light energy. And then you can later consume that sugar. And
fantastic. So the hydrogens in carbohydrates come from photosynthesis.
Yeah, from water.
How interesting.
And this is really important because when we talk about macro scale terraforming Mars, hypothetically speaking, the problem with doing this is that Mars doesn't have enough volatiles. So if you liberate all the CO2 in the ice cap, not enough to make an atmosphere, but it has a ton of water. And like I said, you can use photosynthesis to make oxygen with water. And so the current idea for how to terraform Mars is you convert basically half of the water into atmosphere.
Yeah, okay. We can get into that more when we get a little farther down onto this climate of thread that you mentioned in your title. But just more question about all this stuff. Where's the carbon supposed to come from on Mars?
Yeah, Mars has a lot of carbon. So it has a lot of, it has a, CO2 atmosphere. It has a bunch of dry ice and so on. It has a bunch of, carbonaceous rocks and things like that you can sort of in principle break down. We actually did very early on, we did some like math we went through all the different possible planetary bodies that one might wish to have large scale biomass on. And we computed how many moles of just like carbon, nitrogen, hydrogen, phosphorus, like essential, like non-negotiable atoms, like how much of those things are present on that planetary body. And if they're not there, like how much delta V would you need to obtain it from the nearest source? So this is like really simple, like a very simple analysis, but also very key. If the atoms aren't there, you can't make them, right? Unless you're using macro scale fission or fusion, but we're not doing that. So Mars has the same theoretical upper limit on biomass as Earth. It's actually super similar. It actually has plenty of all these things. They're just at the wrong temperature and pressure.
Okay, and so let's now move forward a bit. So how far have you gotten? I mean, are there organisms that will work in this bioreactor or will then work in the greenhouse? I mean, what do you think? Is this feasible or is this just still at the conceptual stage?
Yes. So things are looking good for the refrigerator-sized lander that makes bioplastic. So we have examples in the lab. We've created bioplastic from simulated Mars dirt, water, and CO2 with an organism that we
and electricity, I presume.
Exactly. Yes. So in this case, we are planning on, instead of having the organism consume CO2 directly through photosynthesis, we're going to use an electrode to convert the CO2 into acetate. And that's an off-the-shelf unit you can buy on the internet. So there's no R&D involved there. But yes, so you would have a solar panel. And that would also do things like stabilize temperature. So
there's nothing wrong with micro fission power plants on Mars, but that's another topic. But that was in Zubrin's original Mars Direct. That was how they're going to make propellant, because you need to split the water, and they weren't using biology. Now, all right, so do you have this refrigerator or parts of it, or where is it at?
Well, right now where we're at is we have organisms that we have made some bioplastic from some organisms from 100% atoms you could get on Mars. The trouble is that relative to how well it would work on Earth, it's worse, right? Because there's toxins like in this mixture that we're giving them. And so what we're doing at the moment is we're trying to apply some directed evolution and engineering to make the microbes better. So we have a starting point. They grow. They don't like it, but they do grow. And that's a great place to be at. So now we just apply a bunch of big hammers
and try to take them from survive to thrive. Right?
Exactly. Yes.
Yeah,
or they're already thriving in the sense they do double in, like, they're not that slow. So that, this would be a process you would do batches every like 2 days or something like that.
I see.
So it's non-trivially fast. But
what is the organism? Is it an algae? Is it a bacterium? What is it?
So interestingly, we tried a whole bunch of things. So we tried a handful of pretty traditional biomanufacturing organisms like Cenocotta and Piputida and stuff like that. We have like 100 different extremophiles in the fridge that are things that have been studied in astrobiology, are things you would sort of expect to tolerate these various stressors. And the organisms that are winning are the traditional biomanufacturing ones. So they don't like the toxins, but the reason we use them in terrestrial biomanufacturing is because they're fast, right? And so I'm not surprised. It's like we're trying to make something that's like a pretty traditional bioprocess. I wouldn't really expect unusual organisms to be better until we get more into greenhouses and outdoor stuff. So I'm not super surprised,
speaking
but
of-
we are trying to do things like move genes from these extremophiles into Cinecot or P. putida, like more traditional stuff, to sort of equip them with some of these extremophile superpowers.
Okay, so I only have a few more minutes before I have to hand it over to audience questions. So let's move into this. I guess I understand green Mars in terms of bringing in photosynthesis and using it, and eventually, terraforming, although you'll start with just making plastic. Where does this whole climate thread come in? And just be forewarned, I'm a bit of a contrarian on most things climate.
Okay. Yeah. Yeah. So, I mean, maybe big picture, I think has happened before, right? So we live on a planet that didn't used to be friendly to human life. It didn't used to have oxygen in the atmosphere. And it was the arrival, the evolution of photosynthesis that converted water into oxygen, oxygenated the atmosphere, and has now allowed humans to exist, right? We live on a planet that has experienced planetary scale chemistry changes as a result of biology. Yeah.
Let me put in a side note on that, because this is something I'm sure you know, but many people might like this if they haven't had this realization yet. So when the poisoning of the atmosphere happened like 3.5 billion years ago or something like that on Earth, because these organisms, these anaerobic organisms, were poisoning their own environment by creating all this oxygen. And eventually, it's rather curious what happened. So organisms evolved that could deal with the oxygen, like us and our ancestors, but also the anaerobes that originally covered the Earth and created the oxygen in the atmosphere, they didn't die. They just retreated to anaerobic environments. And the anaerobic environments include deep underground in the rocks, deep down in the oceans where there's no oxygen, sediments at the bottom of lakes that don't have much circulation, and the guts of animals, which is an anaerobic environment. So it's rather strange that your gut biome largely consists of extremely primitive organisms that evolved without the presence of oxygen, and then had to go find a place where there was no oxygen in order to keep surviving.
Yeah, I mean, it's not even all that far down in the dirt that you get anaerobic conditions. So yeah, it's everywhere still. But yeah, this is all just to say, I think it is, maybe do this thought experiment. If terraforming cost $500 billion, the cost of the interstate highway system, and was going to take 30 years, right? It would probably be actually faster and cheaper to just terraform Mars before we go there. And then it would be to try to go and create a whole human settlement and like have a permanent presence in a climate that is so hostile. Right. And I think the question is, terraforming's not impossible. Like we can't find like a single physics reason why it can't happen. And so the question is, how hard is it? How expensive is it? How long would it take? And indeed, might it be a really valuable alternative to sort of how we've traditionally thought of sort of human society potentially being on Mars? And in the process, of course, I think terraforming as a concept, it pushes on all the right technologies that we should be developing anyway, right? Like we should be figuring out how to make biological engineering that works well for organisms that are robust and maybe don't require incredibly well-controlled conditions and, expensive bioreactors. So that's why I like terraforming. It's a good North Star. It's a good North Star in part because maybe you could do it, right? And that's the inspiring question.
Those are some interesting concepts. But again, I'm going to just poke one more time. You used the word climate in the title of your talk, and I want to know why you chose that word, how you view that concept in this research program.
Yeah, I have a lot of friends who work in like climate biotech. I think there's a lot of interest in finding alternatives to sort of all the technologies we use regularly today. On Earth, it's incredibly difficult for like a new green technology to be adopted, even if it's better in certain ways. And that's because we live on a planet that has literally sometimes hundreds of years of investment in infrastructure like roads and gas stations and like that make it really difficult for sort of new technologies to compete, even if they're the things we'd sort of rather civilization use. And this is what is a real opportunity in a Mars setting, which is we're talking about an environment where humans still need all the same stuff, but there's no infrastructure. There's no status quo to compete with. And so it's this wonderful blank slate opportunity. When we go to Mars, like there's no excuse for not just like making the things exactly the way we want. And so it becomes this wonderful sort of first market for green technology. And I mean that in all levels, like green technology on the level of, can we have a pretty damn close to fully circular, like human habitat, right? Green on one level up of like, could we have human presence in a place have a net positive effect on the surrounding climate of the area rather than net negative, right? Climate, green on the maybe whole planet level of like, could humanity's presence on Mars restore it to a state that it, we think it once enjoyed, many millions of years ago. So that's what I mean by green. Like on
I love it. Regenerative agriculture on Mars. Now there's a climate program I can actually get behind it. Or we could start with regenerative agriculture on the Earth. Anyway, all right, look, we only have about 17 minutes left. So Lydia, maybe you could start asking some audience questions, and I am going to stop talking quite as much as I had, and I'm actually going to briefly vanish, but only for about 1 or 2 minutes.
Okay, great. Have
I
fun.
can still hear the thing while I'm vanished.
Okay, have fun being vanished. I see that Micah had numerous questions. So why don't you go first, Micah? And then if anyone else wants to jump in, they can. And if not, then you can shoot all of them.
Sounds good. First question, what is the theoretical throughput of your hypothetical refrigerator in terms of like plastic mass per day? What do you think is...
yet. We haven't built out a TEA for it yet.
Yeah,
So,
I'm wondering, what do you think is realistic? Like in 10 years or 15 years, what you're kind of shooting for, just broad strokes?
I think we could get to, if we are doing well, we could get to on par with like grams per liter of bioplastic produced. And then the theoretical limit is like, how big is the bioreactor, right? Right now, like organisms grow, or like the amount of bioplastic we can produce is like a little less than half or something like that of like terrestrial yield expectations. So that's the gap that like needs to get recovered. And I think it may also be a little bit slower. So have your batches be shorter and have them be higher yield is like the goal. But I'm not sure. We haven't done a full TEA on it. Your other questions are really good. Maybe just going through those. So we have a Substack where we release technical summaries of like what we've done recently. I linked one that talks about like the infrastructure that we are imagining would be on Mars. Basically, the refrigerator size unit is something that somebody else needs to add dirt, water, and air to, and out pops bioplastic. We think it's pretty reasonable that there is going to be like dirt and ice mining available, because some of those things are now increasingly off the shelf. There's a bunch of like lunar ice mining companies. And in a human mission scenario, basically all human mission architectures that make sense require pre-placement of equipment to generate ascent vehicle fuel, which require ice mining. So I think it's pretty reasonable that like that sort of infrastructure for obtaining those materials and putting them into a box would exist. But it does assume that. And then your last one, this is such a great one. Someone else has some. Oh, it's Danielle. Okay. Is temperature or pressure harder? It's such a good question. The problem is liquid water, which is both temperature and pressure, right? So if you plot temperature, pressure, and you can look at the triple point of water graph, the fundamental problem with outdoors on Mars is that it is below the triple point of water. So if you pour water on the surface, it will either freeze or evaporate like immediately. So liquid water is not stable, and that's the problem. And so you can fix it either direction. You could fix it by increasing temperature, or you could fix it by increasing pressure, or you can do both. But fundamentally, the issue is having it be possible to have liquid water, and which direction you go just changes which organisms you would use, if that makes sense.
Thank you, Micah and Erica. I think Danielle has a question. Would you like to go ahead?
Yeah, hi. Just what I wrote, basically, I'm wondering if the organisms are good enough now to be like placeholders for a mission design. That sounds really exciting. In general, if there's any doubling time to speak of, you'll get there eventually. So how good is it?
Agreed. Yeah. So basically, I mean, yes, I think the organisms are good enough that this idea has legs. And we're actually talking, there's a new sort of ARPA-like funder in the German called Sprindy that we've been talking to, and they want to create a planetary habitability funding program, and they are serious enough about it. They've been talking to SpaceX about upmass cost. So there's like, again, who knows if that happens, but there is like, I think risk in a 2031.
Well, that's really good, right? So I mean, in biomanufacturing here on Earth, you have the problem of any contaminant of things evolved on Earth is much better,
right?
And outcompete something that's getting taxed by your thing. You have the inverse problem with biology you're bringing from your system to Mars. You're not going to get any contaminant at all that's like hyper evolved, or I mean, maybe you could, but then it's a Nobel Prize. So you just send it and see what happens. And a lot of the problems that kill you on Earth are simply irrelevant. Doubling time also irrelevant because you get there. And basically, I see it super asymmetrically, get out in front of this, start raising money for this. Just we're eating it to Mars. We're going. Let's go. That's
totally
my bias.
Okay,
I have
this is like Erica, get back to fundraising. Yeah, no, totally. I think the things I want to see, I think we have some homework to do before that, which is that we can demonstrate growth of organisms that generate plastic. Still a couple different options for which plastic to generate. There's a bunch of devil in the details about downstream processing once you have that plastic, once you have the raw materials. So you have to get it out of the organisms, and then you have to do at least some chemical process to get it into the right chemical purity, essentially, to use it in the applications you want. And that section of what goes in the refrigerator, we have yet to figure out what the best option is for a downstream processing that can be done with entirely in situ resources, right? Because if you have any chemicals, you have to either bring them with you and recycle them 100% or make them on site. So there's so
there's a good amount, but call it months to a year of hard thinking before either you should choose one or just say there's an option A or an option B, and we're, I don't know, crowdsourcing
I think
something.
we'll get to it. I mean, our sort of internal timeline is this is the focus through sort of March, and then we're going to switch focus to thinking more about greenhouse organisms. But yes, I mean, I think we have some good options on the table even now, and it's a matter of like, we have sort of three options: this type of bioplastic, that type of bioplastic, and perchlorate remediation, that in parallel, we're sort of optimizing bioprocesses for all three. I'm guessing one of them will drop out as like the one, the less favored child. And then we'll have probably two at the end of the day that we'd want to make a box that does both, right?
Are you going to make a box, by the way, a control box? Like, wouldn't it be interesting to just have plain old Mars dirt in one of these things, get it into the right liquid water, parameters, feed it the whatevers, and then see what, if anything, just sprouts, if you will?
Oh, 100%. So I mean, and I think a key part of the strategy here is we are planning, like one of the reasons we're not using photosynthesis is less efficient for surface area than a solar panel at fixing carbon. So that's the engineering reason not to do it yet. The other reason not to do it is that we are deliberately using organisms that you have to feed. So they can't grow on their own outside. Like, they are not a planetary issue. Like, they are fully contained because there's no acetate outdoors on Mars. Along with all the others, they would die. They'd die from the UV. They die from the freeze thaw. They're dead if they get outside. But yes, I love it. And I love the idea of every single mission you fly that is de-risking a new type of biotech on Mars. It can double as a search for life mission, right? In which you do exactly what you said. Let's just add water to And maybe we'll put some LEDs in there to see if anything photosynthetic pops out that's sporulated. And indeed, Mars, we have good reason to believe that historically Mars had a climate that looks a hell of a lot like it would have been friendly to life as we know it. And if stuff used to live on Mars, it's probably desiccated or frozen. A very effective style of search for life mission that we haven't tried yet is just thaw the planet or something.
Send the terrarium, micro
Well, I
terrarium.
mean, to some extent, the Viking landers tried that. Like they tried dropping some Martian soil into some chicken broth or whatever it was, and warming it up and see what happens. But another interesting thing, which is maybe more of an astrobiology and anyway, another interesting thing about Mars and Earth and all these ideas for life, extant life or imported life, is, of course, that plants should do okay on Mars under natural light because it has a 25-hour day, which is very curious. Like on the moon, forget it. You can't use natural light for plants because the moon has 2 weeks of sunlight solid and then 2 weeks of darkness solid, and plants can't do that. But Mars has a period. A Mars sol is almost like an Earth day.
True. The light conditions on Mars are a little less than half as bright as on Earth. They're like
For plants.
under the forest canopy style light. Totally fine. Yeah. Amount of light is totally fine. I would say there are a bunch of other sharp corners to navigate, including Mars does have global dust storms not infrequently, which would cause there to be sustained periods where you have much less light. And so, I mean, that's
We'll
Human
solve
plant-
energy storage. We'll just
Human
solve
plants
energy
have
storage
winter.
for you.
totally, yeah.
I mean,
So,
Earth plants in winter.
Agreed. I think the photosynthesis seems viable to me, although maybe not for like indoor bioprocesses that you want to have be very reliable and high yield. And that's why we're not relying on them for that.
Cool. All right. So let's see. Do we have any more audience questions? Because we're almost at the 55-minute mark. Well, I have a couple closing things I want to say. One, I want to thank.
Oh, I think maybe Luke had a question. Sorry.
You got one?
I did. I
Right.
had one more question. Since we're here close to the end, earlier, just kind of nonchalantly, you said, "Oh, we did this great calculation about like take account delta V and the actual elements that are present on the surface of Mars." And we found that it actually has the same capacity to produce biomass as the Earth. So two questions there. The first is, you included delta V. Does that mean you have to like cannibalize Phobos in order to get the elements you need?
Sorry, for Mars, you don't have to get atoms from anywhere. Like literally just atoms already on Mars on the surface. So you don't have to mine anything or anything like that. Yeah.
Amazing. And the
Yeah,
second
but
question,
and then as another fun fact, so like the moon, people keep being like, can you terraform the moon? No, we cannot terraform the moon. Why? Because there's not enough nitrogen. If you mined all the moon dirt for all the nitrogen on the moon, you would get enough nitrogen to make one section of biomass that's like, could be as much biomass as in Yosemite National Park. So like not an insignificant amount on the whole planet, but that's it. And that's not budgeting for an atmosphere, right? So that's like the sort of level of like, and again, these are all very rough order of magnitude calculations, but I think that gives you a sense of like, okay, I see. Like moon, you would have to like transport a bunch of nitrogen to it.
I think you may have actually just answered my second question with the order of magnitude estimate comment, because I was going to say, well, if Mars and Earth are at parity in terms of the amount of biomass that they can produce, is Mars actually way more productive, potentially than Earth? It's so much smaller. It has like hectare per hectare. Like, is it actually more productive than the Earth is?
Depends on, well, okay, the theoretical maximum biomass as a function of atoms. Like, it's not just atoms that determine biomass. So, like, Earth has much less biomass than like, if you converted all the atoms on Earth into biomass, like, we end up having less than that because we have carbon in the atmosphere and we're storing our atoms lots of different places. And doing that estimate is really hard because now you have to really think at a much more granular level of detail about really how do these ecosystems work, right? Where are the atomic resources being stored? So I don't know. I think we don't know like there's a bunch of outstanding questions like, I mentioned that the dirt on Mars is pre-fertilized, so it has nitrogen that is already fixed. So you have like sort of a one-use only opportunity to you convert the nitrogen in the dirt into biomass, and you could get a good amount of biomass. Now, once those plants biodegrade, they release the nitrogen back out into the atmosphere as a gas, and so you have to fix it again, but the atmosphere is thinner, there's way less nitrogen, maybe nitrogen fixation is going to be the thing that limits biomass on Mars, and I think we don't know yet. It's such a different place with
All right,
different
I want to
don't
close it
know.
up because we're almost at the hour. Last tech question: how do Titan and Pluto stack up?
So I was really excited about ice moons because in theory they have all the atoms. The problem is that you have oceans of liquid methane on Titan, which is in theory oceans of bacteria food. The problem is there's no electron acceptor. So there's nowhere you couldn't eat the methane because you need oxygen or something. Like it couldn't combust, right? Like you can't get the energy out of it because there's no other atom to put it on, right? And then on top of that, the ice moons have way less sunlight energy, so you can't use photosynthesis. So they have all the right raw materials, they just don't have an energy source.
All right, great answer.
no.
And I need to thank you, Erika and Lydia and everybody for coming. This is really good. And I want to also mention, this is kind of rare that we do one of these without a half hour or so of prepared presentation, but I think it worked relatively well. I'm open to feedback about that. Anyway, Lydia, want to close us up?
I think you did a great job, but thank you, Erika.
Thank you so much. This was fun.
Yeah. Well, and we'll see you next month.